Hi, Anna. Hello, everyone. We still have a minute left. Here's a random factoid. What makes biology unique? It is the only discipline in the entire realm of the sciences where division is the same as multiplication. Oh. From the standpoint of all biologists alive, the most famous closing sentence in 20th century science, in fact, probably the second most famous sentence in all of biology. It hasn't escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. The closing sentence of Watson and Crick's 1953 paper that not only proposed the double helix you see before you, but immediately suggested that the way the genetic material copies itself is by simply unwinding and then using Watson-Crick base pairing rules to generate a copy. This was tremendously exciting because humanity in its modern form is about 150,000 years old, and heredity, the phenomenon you see here, has been observable by them ever since. But it wasn't until 1953 that humanity had a mechanism to explain why Bianca Jagger's and Diana Ross's daughters look like their moms, respectively. I want to point out, however, there is a very big gap between proposing a generally true notion, i.e. DNA unwinds and then makes strands that look like the parental strands, to implementing it in reality. By way of a specific try to wake you up first thing in the morning example, Jules Verne, the great French author, wrote a book, De la Terre à la Lune, from the Earth to the Moon, in which he proposed how humanity would travel from the Earth to the Moon. And the idea was that the cosmonauts or astronauts or taikonauts, whatever you want to call them, sit in a large bullet. I'm not making this up. And how would this bullet get to the Moon? It would be shot out of a very large cannon. Now, needless to say, the crew of Apollo, Aldrin and Armstrong, are very grateful to NASA for the fact that that was not what was actually done. My point in telling you all this is to say that technical details make an amazing, enabling difference. Here's a mind-blowing fact for you that I discovered in speaking to a Berkeley professor, Nicolas de Monchaux. I don't suppose any of you know what famous American corporation made the spacesuit that Armstrong took the moonwalk in. 3M, no, but good guess. DuPont, good guess. Good for you, somebody said Singer, almost. Playtex. It was sewn by seamstresses at Playtex. They were the only ones skilled enough to sew the spacesuit. Isn't that amazing? This tiny technical difference made possible Armstrong's one step for man, one giant step for mankind. And Professor de Monchaux wrote a book about this, which is remarkable, how Playtex defeated the military-industrial complex. I've shown you this movie before. This is from Dr. Philip Lee, who of course got his PhD here at Cal. It's this marvelous division of E. coli. Incidentally, this bearded gentleman is looking gleefully at what is named after him. This is Theodor Escherich, the German microbiologist after whom Escherichia coli is named. He discovered it. This bacterium can divide in rich medium every 20 minutes, and this requires that its entire genome be replicated. And we will spend today examining the remarkable machine that has solved all the technical details of replicating the entire E. coli genome in 20 minutes. And to give you a sense of the technical challenge, I need to give you some numbers. The first number is Mother Nature has spent 3.5 billion years working on DNA replication. So in non-anthropomorphic terms, of course, and those are more accurate, DNA replication has been shaped into its present state by forces of evolution that have been acting for 3.5 billion years. And the result is, in the Oxford English Dictionary sense of the word, awesome, i.e. inspiring, profound, reverence. By the way, Berkeley subscribes to the OED. It's a wonderful tool if you want to find out what a word really means and where it came from. In principle, here's a bacterium with its circular genome. Incidentally, how do we know that the bacterium's genome is circular? I will show you that in a second. And then replication begins, continues, until two separate molecules are created, identical to the mother molecule, and then they're separated into the two daughter cells. Ta-da! Done! Ta-da! Done. Let's look at the numbers. But before we do that, I need to show you how we know, A, that the E. coli chromosome is circular, B, that replication starts at a specific point. I often point out that the tragedy of the enterprise of molecular biology is it involves the moving of transparent liquids between transparent tubes. It does, but sometimes you get to see. And John Cairns in 1963 realized that if he labeled E. coli DNA with tritium, then he should be able to, if he figures out a way to break the bacterium open very gently, visualize the entire chromosome. Ladies and gentlemen, I give you the entire E. coli chromosome. That's replicating. This exposure took 2 months. Can you imagine doing an experiment and sitting for 2 months going, did it work? Did it work? Did it work? Not for the faint of heart. Now, before you say to me, well, this is just, you know, a circle on a slide, let me give you a sense of scale. Before I dive into this, one thing I was hopeful you would learn is that radioisotopes are really useful in biology. So you learn from Professor Malkin about carbon-14. We learned about Hershey-Chase and their use of S35 and P32. I hope I didn't misspeak in lecture and say that nitrogen-15, which was used by measles and Stahl, is radioactive. It's not. It's actually stable. But it was useful in determining the mechanism of DNA replication. What you just saw is John Cairns marvelously using labeled tritium, a labeled isotope of hydrogen tritium, to show that the bacterial chromosome is one very large circle, how large I'll show you in a second, and that replication likely starts at a specific position in that circle. So the model that we have, that I'm going to walk you the details of, looks like this. Here's a double-stranded piece of DNA. Replication begins at a place called the origin, and then proceeds, and one imagines, and this is the key point, that as replication moves forward, this is very Watson-Crickian. Again, so far, all I've shown you are just cartoons. In principle, this could be happening. I do need to overwhelm you slightly with a bunch of numbers. Do you need to know them? No, you don't. You just need to be impressed by them. So this structure I've shown you before, this is what 12 base pairs of DNA look like. The E. coli genome, this thing, is 4,639,221 base pairs of DNA. So the fact that Cairns was able to get that enormously long, enormously thin DNA molecule to open up for him is quite a technical accomplishment. How fast does this process occur? How quickly does this entity move through the circle? At 1,000 base pairs per second. Per second. And, well, it must be very sloppy because doing things that fast, you make mistakes when doing things that fast. Its error rate is 1 × 10⁻⁻⁻. So it makes one mistake in a 10 billion base pairs. So a bacterium needs to divide 2,000 times before its replication and repair machineries make one mistake in its entire 4.6 million base pair genome. It's an act of unimaginable power. And why is it this good? First of all, it is very, very old. 3.5 billion years of evolution have worked on this. Now, modern humans, let's say ballpark, are 100,000 years old. So in a very simple graph that I'm going to show you here, here's how long old bacteria are. Here's how old humans are. Where is the age of humanity on this graph? It's invisible because it's not a log scale. But just to give you a sense of, we think that anybody who doesn't use Twitter is completely archaic. life has existed for quite a while before the internet, which is why it's so good. It's not distracted. Furthermore, natural selection is astonishingly powerful. Give natural selection this much time, it will evolve a replication repair machine that has an error rate of 1 times 10 to the minus 10. The first challenge before the replication machine is that you unwind one double helix, and this means that you have to make copies of 2 separate DNA strands, as you can see so clearly here, this one and that one. Mission critical fact number 1. DNA synthesis proceeds in the 5' to 3' direction. And if you don't understand what that means, I urge you with all my heart to study vigorously until you do. I explained that slowly last time. This means, as you can see here, that when a given strand of DNA is copied, the polymerase machine, which you can see here, proceeds from 5' to 3'. This also means that the template strand is read in the 3' to 5' direction. Why does this have to be the case? Because the Watson-Crick structure of DNA, in fact, the correct structure of DNA, is anti-parallel. The strands run in opposite directions. So synthesis of one chemically proceeds in a direction that's opposite to the direction in which the other one is read. Let me show you that in specific terms. Now, I will give you letters and numbers, but don't think of them as letters and numbers. Think of them as this pretty double helix. In your mind's eye, rotate that thing in your head. They are not letters. It's what has made you. Furthermore, this is why you can breathe. I decided to use real DNA. This is the DNA encoding the first 6 amino acids of human beta globin. So this molecule is one of the 2 that helps carry oxygen and carbon dioxide in your bloodstream. It's a sad but true fact that 80,000 people, 80,000 of our compatriots, suffer from a condition known as sickle cell anemia. The primary symptom is debilitating pain and acute susceptibility to pulmonary infection and death of stroke. And a mutation in that specific base causes sickle cell anemia. So I will be returning to this piece of DNA many times throughout my section of the class because I want you to know about the predicament that affects 80,000 of our compatriots and many other people on Earth. For now, this piece of DNA shall serve as a model template that gets replicated. By convention, the sense strand is called the Watson strand and the antisense strand is called Crick. What does sense mean? Well, I will have to explain that to you 2 lectures from now. There is a really sophisticated technical explanation for the words sense and antisense. So just learn the word for now, and you will understand what it means 2 lectures from now. Here is the Watson strand, sense. Here's the Crick strand, antisense. Paired, then they denature. And simplifying to one step beyond the truth, and why I say that I will explain in a second. When this molecule replicates, this ought to be happening. Now, in fact, this doesn't happen. So this is a simplification. But I told you that there's a 5' to 3' in this process. So here's 5', here's 3'. The first base laid down will be on this side. That'll be the cytosine. And for this molecule, here is its 3' end, here is its 5' end. The first base laid down will be this one. And the second base, you guessed it. PowerPoint is amazing. And guess where the third base is going to go? Watson-Crick rules are in effect, so you go first one, second one, third one. I'm not going to go through the rest. It's too early for such primitive illustrations. Then we go all the way to the very end, and then the last base gets deposited, et voila. We've completed DNA synthesis. The point of this PowerPoint silliness is to highlight to you the directionality of DNA replication. I also really want to emphasize, this is a deep point and you will only understand it completely in a few lectures. There is no left and right in DNA. There is only 5' and 3'. So this, in fact, means that I could rotate this molecule 180. and everything would play itself out exactly the same way. So chirality in this specific sense, left and right, has no meaning in DNA. 5' and 3' hood does. If you want to build a string of pearls, you take multiple individual pearls and simply put them on a string and you are done. Biological polymers essential for life, however, are very different in that regard. DNA is made from deoxynucleotide triphosphates. RNA is made from ribonucleotide triphosphates. Protein is made from charged tRNA. Triacylglycerols, I made a mistake here. They're in fact made from malonyl-CoA, but they're not made from individual carbon units. Glycogen is made from UDP glucose. The bottom line is, all of these biological polymers, nucleic acids, proteins, fats, sugars, are not made from the building blocks that ultimately form them. So the brick building that is the human body is not constructed from bricks. It is made from thermodynamically embellished bricks. They are chemically modified to make them thermodynamically or energetically easier or more favorable to polymerize. Specifically, once again, DNA is not synthesized from nucleoside monophosphates, even though, of course, DNA consists of nucleoside monophosphates. It's synthesized from nucleoside triphosphates, or DNTPs. What about this? What is this business of the extra 2 phosphate residues? It makes the energetics of the synthesis more favorable. And my point in walking you through all this, again, I apologize. Sleep deprivation is, you're going to say to me, tell us about it, man. It's 8:26 in the morning. It's quite remarkable how you can convert left to right if you're sleep deprived. I'll fix this in the slides before I post them. Sorry about this. This makes the energetics of the synthesis more favorable, is what's going on. Let me distill this to specifics. Here is the elementary chemical act of heredity. One chemical reaction to generate one additional base pair. Here's the template strand, 3' to 5'. Here's the new strand, 5' to 3'. This base pair exists, this base pair exists, this phosphodiester bond is in place. This base pair is a thymine. An adenine needs to go in here, so an adenosine triphosphate, deoxy, DATP, is brought in, and then this chemical reaction takes place to form the phosphodiester bond. And as I will explain in a single slide, the release of this pyrophosphate and its subsequent hydrolysis provides wonderful thermodynamic impetus for this reaction to actually go forward. But before I go into that, I realize that this is somewhat dry. I want to get this to be as real world as it humanly can be. I'm wondering, by show of hands, how many of you know who this is? I'm not going to call on you. I just want to see sort of how big the generation gap is. Okay? Would somebody care to tell me who this is?
Patrick Swayze.
This is Patrick Swayze. He was diagnosed with pancreatic cancer in early 2008 and died of it in mid-2009. I believe there are about 250,000 cases of pancreatic cancer in the world every year. 30 or so thousand people of it die every year. It is probably one of the 2 most lethal cancers known. I believe that the 5-year survival rate for metastatic pancreatic cancer is somewhere around 3%. By comparison, the 5-year survival rate for breast cancer, if detected early, is over 80%. Patrick Swayze's life, and those of many other people who don't want to just die, was prolonged by a chemical known as gemcitabine. It is, in fact, the standard of care for pancreatic cancer, as well as in combination therapy breast cancer. There are currently 1,316 clinical trials using this molecule. And why so many? Because it's the standard of care. So if you want to improve and help people live longer, you have to use drugs in combination with it or compare to it. And why am I telling you all this? Homework, friends. This is the chemical structure of gemcitabine. It is, in fact, 2' deoxy, 2' difluorocytidine. By way of homework, or conversation with your GSIs, or your physician parents, or your chemist parents, or Facebook. Put a call out on Facebook. Why is this drug effective in killing cancer cells? Specifically, why is this drug so effective in killing rapidly dividing cells? Let me give you a hint. It is sitting on this slide, the slide that shows the biochemistry of DNA replication, for a very good reason. Its mechanism of action, and this is where the hint ends, its mechanism of action has very much, in fact, has everything to do with what you see in front of you. And I walked you through this example to highlight to you that whatever your future, physician, public health professional, or human being, there is the non-zero possibility that you will meet this molecule, directly or indirectly. Now, from this, I want to go back to very basic principles of the universe, which is the second law of thermodynamics, and it cannot be violated. It's a law. Says, in very introductory level terms, that the level of disorder or chaos of the universe always increases. Now, those of you who want to take MCB 102 or upper division MCB classes will have to learn a proper definition, but that's for aficionados. DNA replication, therefore, is the generation of order from chaos. So, on first principles, would appear to violate the second law of thermodynamics. Imagine that you have to replicate these, however many, 18 base pairs of DNA. So you need to synthesize this thing in green in its entirety. What do you need to do? Well, you need to gather from the cell 6 DGTPs, 40 CTPs, 6 of these, and they're just happily floating, disordered and excited. And you need to put them together in that ordered string. In principle, this appears to be violating the second law of thermodynamics. The universe, however, is very smart. And the way it solves this problem is, in fact, via this little step. And I wanted to highlight to you how sharp the universe is. Well, the universe doesn't have intelligence yet. How worthy of admiration the universe is. How about that? Specifically, it doesn't violate its own laws. If you have DNA and you polymerize it with a deoxynucleotide triphosphate, you get DNA that is longer by 1 nucleotide, as you just saw, plus this entity, P-P. Look up on the previous slide what the technical name for that entity is. What that previous slide correctly points out is that that entity is then hydrolyzed to form 2 simple inorganic phosphates. So you start with 2 molecules and you end up with 3. And here's a picture of the universe excited about this. That's how I visualize the universe, glad over the fact that its laws have been obeyed. Continuing this anthropomorphic silliness, the chemists and the biochemists and the structural biologists who study DNA polymerase have decided to get cute and to declare that DNA polymerases have a thumb, a palm, and fingers, and thus, in fact, that DNA is held by DNA polymerase like this. And in fact, this is the drawing from your book. This is a picture from a review, and the purpose of this color coding scheme is to simply highlight that the thumb, palm, and fingers arrangement is preserved in polymerases from unbelievably diverse walks of life. It's just a very old idea that evolution liked and stayed with it. I want to actually show you briefly what this looks like. I don't want to spend too much time on this, and I'm not really expecting you to memorize this in in any way. I just think it's marvelous. So this is DNA polymerase, a DNA polymerase. And here is, I believe, thumb, palm, and fingers. And if you zoom in, here is a DCTP with its triphosphate, and here's the previous nucleotide, and the elementary act of incorporation is about to happen. There it is, high-resolution structure of the elementary act of heredity. What would I like you to know about this? Simply that DNA polymerases have a very conserved structure. It's an old solution, one that Mother Nature has used again and again. What I'd like, in fact, to spend a bit more time talking about is a remarkable accessory, an accoutrement, that DNA polymerase comes with. It's not just a machine for synthesizing DNA. It's a machine for synthesizing DNA with high processivity, which means once it starts, it keeps going and going and going and doesn't let go of its substrate. How can it do that? It can do that because DNA polymerase comes with a clamp. There you go. It's a ring. Now, it so happens, again, by way of Go Bears, a little bit of blue and gold pride, that the clamp was discovered by, and its foremost scholar on Earth is, in fact, Berkeley's own Professor John Kurian. And I thought it would be appropriate to basically read from his writing, because I really cannot improve on the man who discovered the thing. The raison d'être for the clamp is the faithful and efficient copying of chromosomal DNA is performed by chromosomal replicases, which generate double-stranded DNA from primed, what that is I'll explain in a second, single-stranded DNA. These replicases achieve great speed and processivity by tethering to ring-shaped protein complexes called sliding clamps, which encircle DNA and have the ability to slide freely along the DNA strand. The DNA polymerase subunits of these replicases, these fellows, maintain their association with the template strand by remaining attached to the clamp, which moves along with the polymerase during DNA synthesis. The E. coli replicase, DNA polymerase III, is one of the best studied chromosomal replicases, and this is actually kind of cute. It has been shown that in the absence of the sliding clamp, the replicative polymerase cannot synthesize more than a few bases without falling off. Oh, ah! As opposed to, it's remarkable. Before I show you the next structure, I need to show you a cartoon and a movie. And then I will show you some very deep biology. So the purpose of the next 90 seconds of complete silliness is just to wake you up. Don't take any notes, just watch. As you can tell from my name and accent, I was born and raised in Russia. And as a kid in Russia, you see these. These are bears in a pine forest. And aren't they cute? And then many years later, I visited the Healesville Sanctuary in Australia, next to Melbourne. And I saw a bear. Just kept going. There were like 20 people around it, oohing and eyeing and saying things to it. And it's just like, look, man, this is lunch. Get away from me. Isn't that amazing how ursine it is? Or if I were to switch from Latin to the Greek, how arctic it is. Arctos is Greek for bear. Now, needless to say, the koala is not a bear, even though its genus name has the Greek word for bear. It's a marsupial. Everybody knows this. Elementary school children know that. This is a wonderful example of analogy. Similarity that does not reflect evolutionary ancestry, commonality of. The many similarities between Ursus arctos, which is Latin and Greek for bear, and the koala are not due to common ancestry. On the other hand, of course, there's conclusive evidence that the similarity between the domestic cat and the lion is, of course, due to descent from a common feline ancestor. Escherichia coli, the bacterium that lives in our gut, and Homo sapiens, the human being who sometimes thinks, both have a sliding clamp to ensure fidelity of DNA replication. They do not have the same amino acid sequence. They did not evolve from a common ancestor. They evolved independently. And what I'd like to show you is something completely breathtaking, which is this is the bacterial one. Whoa. Look at that. And this is the human one. Mother Nature just liked this solution. She was like, a clamp, processivity, fidelity of replication, must evolve again. So analogy is observable macroscopically. The dolphin is not a fish. The koala is not a bear. And on a molecular scale, the DNA clamps that replicate the bacteria in our stomach and in the lining of the stomach are analogous to each other and do the same thing. Okay. Now, I am a very bad artist. I'm sorry, but I could not find a diagram that would show what I need to explain to you, so I had to draw it. Apologies for the fact that this looks ugly. The anti-parallel nature of the double helix and the strict 5' to 3' rule for DNA replication creates a problem. So this is starting DNA. Let's unwind it and replicate it. And this one. Now unwind it some more. Oh. How are we going to replicate that? We can't use this polymerase because it's about to fall off this end. Go, whee! This problem is solved using a remarkably elegant solution. In brief, the biochemistry of DNA replication involves, if the DNA is being replicated this way, so this is the replication fork, a leading strand template, which is just replicated, and is thus engaged in continuous synthesis, and a lagging strand template, a term I need you to memorize, lagging strand, which engages in discontinuous synthesis. The product is still one chemically conjunct double piece of DNA, but it is synthesized discontinuously in a beautiful simile that was provided actually by Arthur Kornberg, the father of the study of DNA replication, is the lagging strand is replicated in staccato fashion. Specifically, by the way, the next sequence I need you to know for the midterm. What I need you to do is to watch the webcast and play these slides on your screen and more or less recite along with yours truly until you can recite this from memory. Now, I'm not expecting you to memorize it. I'm expecting you to understand all of this, okay? So, let's go. We have DNA being unwound. This is the direction of replication. This is the replication fork. We begin with an enzyme called a primase. It synthesizes an RNA primer, ribonucleic acid. DNA polymerase can then engage those RNA primers and use them to synthesize DNA. Thus, we have the leading and the lagging strand. Okay, we're done. We've made a copy of this chunk. What happens now? This thing and this thing come into play. A DNA helicase will unwind the DNA some more, and topoisomerase will relax the superhelical stress. What that is, I will explain next time. We're going to unwind more of this in this direction. And what was going to happen there? Well, first and foremost, as more single-stranded DNA becomes available, DNA polymerase and its incredibly ancient clamp can proceed to synthesize the leading strand in continuous fashion. As I showed you in my rather silly drawing, we have a problem in that we've generated a gap that needs to be somehow replicated. Not to worry. Primase produces a newly synthesized primer. It will be extended by DNA polymerase. And what you now realize is on the lagging strand, we have generated two short RNA-DNA hybrids. And those hybrids are immortalized. I take that back. The man who discovered them died at age 45 of leukemia. He fortunately lives in the fact that the fragments he discovered are named after him. His name is Reiji Okazaki. That's an Okazaki fragment. That's one, and that's another. Our goal was not to synthesize short, pretty fragments. Our goal was to synthesize a continuous DNA strand. A separate DNA polymerase and other machines remove this RNA, synthesize over this gap, and then there's a nick. So there's a missing phosphodiester bond. That phosphodiester bond is healed by a DNA ligase. And that, folks, is the complete sequence of DNA replication, leading and lagging strand. So what am I expecting you to know? One more time, I'm expecting you to know the dynamics of the process and the functions of the individual players. Aha, the quiz. Take out your clickers, please. Now, students mentioned to me that some of them could not gain a signal. I think, I'll give you 2 minutes to do this, I promise. I will give you time to do this properly. Spend 10 seconds thinking about this. And I also constructed this device with very long cables such that the sensor is visible to you. If you push the answer button once, then you should get a green light. And if you get a green light, your answer has been recorded. Okay, so I'm not asking you anything yet. Don't panic. Let's start this session. What does this mean, searching for bases using the same phrase? Aha, okay. All right, folks. One of these statements is wrong. Which one? The directionality of a single strand of DNA, such as the one you see here, is a function of the fact that the phosphodiester bonds connect to chemically distinct carbons in the deoxyribose. 2, a useful feature of DNA from a function as genetic material perspective is that it can accommodate any sequence of bases. Any one of 4 bases can be chemically linked to carbon 1 of deoxyribose. 3, each base is near perfectly planar when viewed from the side, of course, and this accounts in part for the beautiful spiral staircase structure of double-stranded DNA. 4, each base has unique properties with respect to its ability to engage in chemical reactions. And these properties, the chemical reactivity of the bases, of course, determines the rules according to which base pairs form when the 2 strands of DNA come together. Finally, while both purines and pyrimidines can occur on the same strand of DNA, and they actually have very different sizes, as you can see here, the base pairs have very similar geometry. And as a consequence, the DNA double helix has a highly regular structure with no bulges in it. Okay? One of these is wrong or inaccurate. I'll give you a full minute and a half or however much you need. I'll start the session. Don't rush. Take your time. Exhale. It's just a quiz. By the way, feel very free to talk to your neighbors. All right, so let's take another 45 seconds, if that's okay with you. And then when the timer says 2 minutes, let's stop and move on. Did any of you try to use the clicker yet?
Oh, yeah.
All right. Wow, 540 people. That's really impressive. This is essentially the capacity of this auditorium. 45, 46, 47. And the artist wins. Okay. Thank you. All right, folks. Brace yourselves. Wow. Cool. Okay. I promised myself that if more than 20% of the answers were wrong, then this meant I didn't teach you this properly, and it would be unfair of me to test this on you. And therefore, because the incorrect answer is D, so the 38% of you who said D, nice job, I'm very, very impressed. This was a tough one. I realized that maybe I didn't teach you this well enough, so I need to teach you this to you again. So this point is canceled. Don't be sad. You will have ample opportunity to succeed. It's not about points, it's about learning. All right. So let me explain what's going on. Yes, of course, this explains the fact that DNA is directional. Yes, DNA can accommodate any of the 4 bases. Yes, each base is near perfectly planar. I showed this to you. Yes, the base pairs have different geometry. But what I guess I didn't explain to you well enough, but I'm about to, is that the double helix, the formation of the double helix, is not a chemical process. There is no chemical reaction. It's hydrogen bonding. No serious chemist would call the breaking and the formation of a hydrogen bond a chemical reaction. It's a transient quasi-electrostatic interaction. So the bases themselves remain chemically inert. This is the remarkable thing. They just project from the outside perimeter of their ring a pattern that says, I am an adenine. I'd like to pair with a thymine. And the cytosine does the same. I'm a cytosine. I'd like to pair with a guanine. And guanine and thymine do the same thing. That's when they come together, they basically project these recognition codes until they dock with each other. So the reason that statement D is incorrect is statement D said, oh, you know, there are these chemical reactivities. It's not the chemical reactivities. It's the pattern of hydrogen bond donation and acceptance. And in thinking about what question to ask you, I realized that maybe I didn't spend enough time on this fundamental process through which Watson-Crick base pairs form. And I decided to test whether or not that's true. And as this experiment just showed, that is. The 38% of you who got it right walk from class going, point. And now for something completely different. Another remarkable person who got his PhD from UC Berkeley, the inventor of PCR, Kary Mullis. I'm not going to spend any time explaining why the slide background looks like this. If you want to investigate this on your own time, be my guests. I do want to emphasize that the revolution in biology that we've lived through is due to recombinant DNA technology, due to all the people you see here, DNA sequencing, which is due to Fred Sanger, and there's nothing to discuss. Kary Mullis invented the polymerase chain reaction, and to this invention we owe many remarkable developments. In fact, I certainly wouldn't be here without PCR. I'll give you a very specific example of what you would do today as a physician or a molecular geneticist or as a public health professional if you were in the field today. Remember this chap? The one who was treated with an adeno-associated virus and was cured, or well, quasi-cured of his congenital blindness. The disease that he has can be caused by mutations in one of 16 different genes. But the drug he was treated with was tailored to mutations in just one, RPE65. So today, if mom visits a physician and says, what's a physician? A physician, and says, oh, Dr. Lee, I saw this story about a cure for Leber's congenital neuropathy. My son has LCA. Can you help him? And Dr. Lee immediately has the boy genotyped, uses PCR to determine if his mutation is in the RPE65 gene or not. If yes, he can be treated with that specific drug. If no, he can't. And this takes 36 hours, of which much is the waiting for the result of sequencing. 30 minutes to get the DNA, 2 hours to set up and run the PCR, which of course by now you should have done. 24 hours to get the results of sequencing back, and 30 minutes to analyze the sequence. It's a remarkably powerful diagnostic test. Why is this so fast? Well, let's do some elementary math. Let's say you start with genomic DNA from 5,000 cells. That's 10,000 copies of an actual gene target. After 30 cycles of PCR, you get a microgram of a given fragment. Oh my God, be still my heart, a microgram. A 100 base pair PCR product weighs that much. This means that a mole of it weighs that much. So 1 microgram that you just got after 2 hours of work, well, you didn't know work, you just stuck a tube into a PCR machine. You get that many moles, and that many moles contain 9 trillion molecules. PCR allows you to go from negligible amounts of DNA to that many of just the region of interest, which you can analyze in single-digit hours. And that's just one application. I walked you through specifically LCA because, in fact, Mullis was working on diagnosis, on diagnostic kits for genetic disease when he invented PCR. And I don't need to walk you through the slide because, of course, you've had a wonderful lecture about this from Professor Meehan, and you've done PCR. So I don't want to focus on the enzymology. I want to focus on a specific application. Bear with me for 3 minutes. This is worth your time. I do want you to compare and contrast. I think this would be useful for you by way of a learning experience. Cellular DNA replication and PCR. What's different, what's similar about them. And that, of course, is why I teach them side by side. I only have 3 minutes. There is no way for me to teach you this to you properly in 3 minutes, so I'll just talk very, very fast. This will not be on the midterm, but this is the human karyotype, 46 chromosomes. Somewhere there is the genetics of human identity. In principle and in practice, the complete DNA sequence of a given human being identifies that person unambiguously. Logistically, it's very expensive and time-consuming to sequence the entire genome. So this is all from John Butler at NIST. Something tells me that somebody in this room at some point in their lives will become involved in crime solving, or heaven forbid accident victims, and heaven forbid soldiers at war, paternity testing, inheritance claims, the money is mine, missing persons investigations, and heaven forbid, and this stuff. But these are all the applications of use of PCR in human identity testing. I just told you that the goal of genetic identity testing is to find what makes a person unique. But what was practical is not to weld a titanium spacesuit, but have seamstresses at Playtex sew them. That was the best option. For identity testing, what is read out in the DNA sample is not the piece of DNA that's responsible for eye color. What's read out is a biologically unimportant barcode that each of us carries in our DNA as a result of an accident. On 13 different positions in our chromosomes is a molecular barcode, the famous CODIS loci. And your textbook explains that the barcode consists of repeats of the same short DNA. And what's different from person to person is the number of repeats. Bear with me for 60 more seconds. It will get better very soon. So you can do PCR to measure for any given of the CODIS loci, whether you have 12, 14, 16, or 17 repeats. And the point is, for all of us, the number of repeats in the CODIS loci are different. 13 loci have been selected. In order to determine if DNA comes from person X, you simply amplify them all by PCR. Now, your textbook shows this. In fact, what is done is actually pretty remarkable. You basically do a 13-fold multiplex PCR reaction in the same tube at the same time. And for every single one of the CODIS loci, you measure the size of the PCR product. And that, folks, is the complete genetic fingerprint of a human being. It's converted into a number. And the number is then put into a database. And then you can use the product rule to determine whether or not this DNA sample is a match to the suspected father. Why you can use the product rule, Gregor Mendel will explain to us. Just one example, and with that I'll let you go. You Google News this morning, DNA evidence. This is a tragic story. Suspect pleads not guilty in Kathy Taft's death. This person was charged in the March 2010 rape and death at a home and rally. Defense attorneys asked the judge to throw out DNA evidence. Attorney Michael Driver said police should have obtained a search warrant before picking up the cigarette butt. This is how powerful PCR is. You can get genotypable amounts of DNA to do this off saliva that was left on a cigarette butt. The judge rejected the defense request. Whoa. Office hours, 180, 10. Now.